Welcome to Chapter 4, Cellular Membranes and Transport. Under that, we will be starting off with subtopic number 1, the Fluid Mosaic Model of Cellular Membranes. Okay, a few key pointers I would like to give you about the cellular membrane. Now, since your small days, you have been able to denote what a cellular or cell membrane is. I think a noteworthy mention would be that cell membranes are also known as plasma membranes. More on that later. Now, the cellular membrane has a specific width. This is not exactly the same across all of the cell membranes that you're going to come across in biology, but this is a generalized width for all the cell membranes. We say that a usual cell membrane is 7 nanometers wide. An average, a typical cellular membrane is 7 nanometers wide. You will find nanometers 8, you will find nanometers 9, you'll find 10 nanometer cellular membranes. It's perfectly fine. But typically, a cellular membrane is considered to be 7 nanometers in width, right? And then cell membranes are composed of something known as a phospholipid bilayer. This is the most important aspect that we'll be further discussing. Now, bi means two. Bilayer means two, meaning two layers. I'll be touching upon that a lot. Now, cellular membranes, they separate the cytoplasm of the cell from the outer environment. And cell membranes are very crucial to control the exchange of materials. Exchange of materials between, that does not look like a between the cell and its surrounding. You'll understand all the different mechanisms that are going to be utilized by cell membranes to make sure they control the exchange of materials between them and the surrounding. Now, why is this topic like this? Why we call it the Fluid Mosaic Model? It's not just out of spite. There's a specific reason why this is called the Fluid Mosaic Model. Right. Here's the generalized idea. Whenever we take a general cellular membrane, we say that any cellular membrane is both fluid and mosaic. Hence the derivation of the name, the Fluid Mosaic Membrane. Okay, then what does fluid and mosaic mean? Let me pick a good color. I think I want to go for a purple. Right. The first one, let's take it step by step. First, what does fluid mean? Now, the cell membrane is made out of a bunch of lipids known as phospholipids. More on phospholipids in a bit, but simply the idea of the fluid mosaic membrane being fluid lies behind this. Now, these phospholipid things and some associated proteins can move. Can move about the monolayer by diffusion. That should make no sense to you, and that's perfectly fine. Just hold on to that thought. Now, why do we call it as being mosaic? Just consider general life. If you consider general life, Mosaic means scattered, right? Mosaic art, mosaic pieces, scattered, scattered, scattered. That is the exact same notion when it comes to cell membranes. The scattered proteins. Now, around a cell membrane, we have many proteins. I'll visualize all of this in a bit. The scattered proteins on the bilayer produce a mosaic-like structure. Produce a mosaic structure. Okay, now to put all of this into effect, let's draw our first cellular membrane comprehensively. Which color should I use is the next problem. I'm going to use another shade of green. Okay, this should be fine, right? Now, we know what a phospholipid looks like, right? It has a head and two tails. Coming from our biological molecules chapter, we should know that a phospholipid has a head and two tails, right? Now, I'll draw the first layer. Okay, this is the first layer. Right. Now, we specifically mentioned how this was actually a bilayer. I'm not doing anything, I'm just adjusting the tail sizes. Give me a second. Okay, right. Now, we discussed that this is a bilayer. The cell membrane is a bilayer, meaning not just one layer. So, this I've drawn here, this is just one layer. This is a monolayer. It's not a monolayer. The cell membrane is a bilayer. That's the idea. That's the idea. There's one layer here, there's the second layer here. It's a bilayer on both ends. It's a bilayer on both ends like this. And scattered among this, you'll find different variants of proteins. I'll draw one protein. So here, spanning the bilayer, I have a protein that looks like this. Okay, and then we can just continue the phospholipid bilayer. So 1, 2, 3, 4. I'll draw 4. It's a bilayer, remember, so it has to be on the other side as well. These are the tails of the phospholipids. You don't have to perfectly draw them. That's not an issue if you don't draw them perfectly. It's just the idea. Now I'll draw another protein. So this is something known as a channel protein. I will discuss about all these specific types of proteins in more detail. So it's another protein, and we can continue the bilayer again. See, everything is just scattered within the bilayer. That's why we call the bilayer as being a mosaic model. Right. Let me centerize things a little. Now I have a bilayer with different proteins embedded onto it. So let us move it down a little bit. Now I'm going to start labeling things. Okay, we'll start with what we already know. This is simply a phospholipid. This is one layer. This is the second layer. Both of them together make up the bilayer. Okay. Both of them together make up the bilayer. Now, what about this? This is simply a protein. Alright, this is simply a protein. Very simply. Now, I'm going to connect this protein like this to something. That is simply a carbohydrate piece. Okay, the protein is connected to a carbohydrate piece. Why is this done? That you will understand once we go more into depth when it comes to this chapter. But for now, all you have to know is that is a carbohydrate piece. When the carbohydrate piece is attached to the protein, we say that this is a glyco for the glucose or carb piece and protein for the protein piece. This is a glycoprotein. This is a glycoprotein. What if I attach another glyco piece onto a lipid like this? This can also be done. Now what I have here displayed is a glycolipid. Is a glycolipid. Is a glycolipid. This is what is known as a channel protein. More on that later, but for now I'll explain why it's known as a channel protein. Now if I have a certain molecule on the outside of the cell, let's imagine this is the outside and this is the inside of the cell. If I have a specific thing, if I wanted to bring this in, here's the deal. Some molecules can move through these phospholipids straight away, not an issue. There is a specific segment later on about transport. Once we get to that, we'll discuss about the specificalities of molecules, whether they can cross or can't. Now, if they can't, if the molecule cannot cross through this, it has to go through special channel proteins. This is almost like a hole. It's a small pore, we say a pore, that molecules can go through to enter the cell. So if I zoom out and help you visualize this, this is going to be the integrity of the cell. So we have the cell membrane like this. That's the cell membrane. This is the outside. Things are coming in from the outside. We have the nucleus and everything inside here. That's an extended version of the cell membrane. Now, you know, when we draw cell membranes, we just do that, no? But in reality, if you really zoom in, now we know it's a bilayer. It's a bilayer. We have two layers to it. I get rid of that and that, also that and that. Okay. So going back to this, these are roughly the main parts that you're going to find in the cellular membrane. I have omitted one specific thing. And that specific thing is something like this. So it's going to sit in between the phospholipids like this, in random places of the bilayer, like so. And that is cholesterol. We have cholesterol molecules specifically scattered throughout the membrane. Why? We'll discuss that later. Now these proteins, let me pick an orange color. Yeah, that's better. Now these proteins are typically referred to as intrinsic proteins. Why are they known as intrinsic proteins? That's because they are embedded within the bilayer. See, this is the bilayer and they're embedded within the bilayer. So you call them intrinsic proteins. And at the same time, you can have simple proteins attached to the cell membrane on one side, on just one side. And we usually call them extrinsic proteins. Extrinsic proteins. We sometimes also refer to them as integral proteins and intrinsic proteins. But the idea being, if it's fully embedded onto the bilayer like this, you call it an intrinsic protein. If it's just attached to one side, it's just basically an extrinsic protein. With that, here you have all the random structures that are going to be needed to learn about the cellular membrane and study its function in the upcoming series.